All right. Good afternoon and early evening, and welcome to the third of a series in this SOARCE webinar series, which is Sharing Ocean Acidification Resources for Communicators and Educators. The title of today's presentation is Building Ocean Acidification Research, Bringing Ocean Acidification Research to the Classroom, a Systems Thinking Approach. This series is hosted by NOAA National Marine Sanctuaries and Ocean Acidification Program in collaboration with many others, including Washington Sea Grant and the Institute for Systems Biology. Our goal is to provide ocean acidification communication tools to formal and informal educators and stakeholders across the country, and to promote a more integrated and effective ocean acidification education community. And I'm very pleased to report that we have 205 people registered for the webinar today, so thank you all for joining us. I'm Laura Francis, Education Coordinator with the NOAA Channel Islands National Marine Sanctuary, and I'll be facilitating the session today. We also have Jen Bennett-Mintz, Education and Outreach Coordinator for the NOAA Ocean Acidification Program online. She's a co-organizer of the series, and she'll be helping to answer questions from attendees. So during the presentation, attendees will be in listen-only mode. You're welcome to type questions related to technical issues or questions for the presenter into the questions box, and we'll be monitoring incoming questions and we'll respond to them directly, or we'll pose them to our speaker. And we're also recording the session, and we'll share the recording with registered participants via Google Drive. So to start things off, I'm going to go ahead and we're going to get start with a poll and see which group you self-identify with. And so if you could answer the question in the poll. %, voted, so we'll give it another 30 seconds or so. Okay, I'm going to go ahead and close the poll and share the results. So it looks like we've got a lot of informal educators, about 49% informal educators, 25% college educator or scientists, 6% formal educators, and 4% community members. Thank you very much for responding to that poll. And now it is my honor to introduce our speaker today. We're very excited to have Claudia Ludwig, Education Program Manager at the Delegates Lab, Institute of Systems Biology, and board-certified teacher presenting today. Her research focuses on instilling higher-level thinking skills in students through systems biology research. To accomplish this, she establishes interdisciplinary teams of scientists, engineers, teachers, and students to learn about current systems research. The team then translates that research into curricula, activities, lab kits, and training experiences, including the module on ocean acidification that you're going to hear about today. Claudia graduated from Loyola University in Chicago, earning a bachelor's degree in biology and a master's degree in education, with additional emphasis in chemistry, curriculum, and instruction. She also currently serves on the Puget Sound Biotech Advisory Committee. So I'm going to go ahead and hand the screen over to Claudia. And we can go ahead and get started.
Okay, thank you very much. I'm excited to be here today talking to you about how you can bring ocean acidification research to your classroom, and how it seems from so many informal educators here today, how you can help others bring ocean acidification research to classrooms and different experiences, and specifically how you can use a systems thinking approach to teach that ocean acidification content and research. And what I'd like to do today, let's see, my slides are not advancing here. Okay, what I'd like to do today is spend just a short amount of time talking about our program that developed this curriculum, a little bit on ocean acidification. I'll give you a broad overview of the module lessons and how they are generally implemented, and then we'll spend the majority of our time on the specifics for implementation. And I'd like to reserve about 10 to 15 minutes at the end for questions that you may have. So first, just a little on our program. I work at the Institute for Systems Biology, and what we are is we're a nonprofit research group using systems biology. And what that means is we study systems holistically, and we do that to increase wellness for humans and the planet. Our mission is to revolutionize science and be able to do more of these holistic studies and to transform human health and ensure environmental sustainability. Within the Institute for Systems Biology, we have 9 different research groups, and I work in the Delegates lab, and here we are. And what we are is we're interdisciplinary scientists and educators, and we use systems biology to study the interplay between the environment and model organisms. And within that research, we have an education effort called Systems Education Experiences, where we translate that research and teach it to high school students, undergraduate students, and teachers so that we can cultivate cross-disciplinary skills for solving complex problems. The program has been running for about 10 years, and thus far we have developed 4 curriculum modules. The module I'm going to talk to you today about is the Ocean Acidification module, but specifically all of our modules are interdisciplinary, and the reason they're interdisciplinary is because our research is interdisciplinary. We do science, technology, engineering, and math in order to look at the big picture of all of these issues. They're all very hands-on and inquiry-based, and all of our modules are connected to current standards. So for this module in particular, that means it's connected to Washington State standards and then the next generation science standards as well, and we'll go over that a little bit more when we talk about implementation. They're all online and free to use. They've all been developed through National Science Foundation funding and made available to teachers and formal educators and anybody who is interested. Now, a lot of times when I talk to people about our curriculum, they question how it can be interdisciplinary and yet taught in a biology class or in a chemistry class. And the way that we do that is that all of our modules are based on some common problem that is inherently interdisciplinary. And then it's the teacher's job to bring in the specific interdisciplinary content as is pertinent for their students. Also, all of our modules are focused on thinking. And what that means is that we walk students through the process of research, and we help build higher level thinking skills. And what that means to us is that they're able to solve complex problems, they're able to be innovative and creative, and we walk them through that process and scaffold that learning so that they can become better thinkers. Sometimes when people first come to our curriculum, and I can give you an example of our gene networks curriculum, is I've had teachers say, well, I went through the gene networks curriculum, but there's nowhere that you really describe what a gene is. And that's true. And the reason that we do that is because in textbooks you can learn very easily or very nicely what genes are. But what's hard to do from textbooks is figure out how you can study genes, how you can study the interplay between the environment and genes. And what we do is we focus on that thinking. All of our modules are also widely used in high schools. So we have 15 different states using our modules and thousands of students. And we mentioned that they're used in high schools, but they're also often adapted for middle schools and for community colleges. Now, I want to go over just a little bit on the case study, but before I do, I'd like to set up another polling question so that we can query your understanding on where you are with your current ocean acidification. So, Laura, were you able to set up the polling question? Oh, there it is.
The poll's up, and we've got about 73% voted, so we'll give it another 20 seconds or so.
Perfect.
Okay, I'm going to go ahead and close the poll, and I'll share it, so you should be able to see it. So it looks like we've got a big group in somewhat familiar, mostly familiar, and very familiar.
Okay, so it seems to me that what we can do, I think our best steps at this point is to move through the curriculum, and what you'll see is there'll be several points within the curriculum that I'll give you a clear overview of ocean acidification. And just for that 5% that are unfamiliar, so ocean acidification is the process of carbon dioxide from the atmosphere landing in the water and beginning a series of chemical reactions that decreases the pH or increases the acidity of the water. The ocean is buffered, but with all the CO2 that has been going into the oceans now for many years since the Industrial Revolution of this happening, we're beginning to see changes. And so these lessons will actually introduce that to students, and throughout the webinar we'll be able to learn more and more about ocean acidification. Now, Dr. Monica Oriana is the scientist in the Delegates lab that first came to me about this particular issue. So in 2009 she came to me and started talking about the growing evidence that human release of carbon dioxide is having this profound impact on marine ecosystems. And she really felt that it was crucial for today's students to fully understand because it's happening now and it's creating changes and problems now. It's a scientific problem, but also it has these largely anthropogenic roots. And this results in serious biological and societal consequences. And so when we started talking about this problem, what we wanted to do is build in an opportunity for students to not only act as scientists, but to also act as delegates, since there is such a human component to this particular phenomenon. Also, we realized that the science of ocean acidification is a nice opportunity to bring inquiry-based learning and experimentation into the classroom. There's a lot of research happening that is difficult to replicate in high schools, but ocean acidification research can be replicated in high school. We can closely model what's happening so that students can be learning real-time what's taking place in our oceans and around the world. We also realized that this was a wonderful opportunity to connect to the work of others. Ocean acidification is complex and there's a lot to do to understand it. And by working together and connecting to the work of others, we've been able to build on our expertise and also connect to the work of others. Our expertise is focused on phytoplankton, which is the plants of the sea that release an incredible amount of oxygen that we rely on. And so while that's our focus, we build on the work of other people to bring in a nice holistic view of ocean acidification with this particular module. So let's look at the lessons at a glance before we go into the specifics for implementation. So the main goal of the entire module is to have students analyze the effects carbon dioxide has on ocean chemistry, ecosystems, and human societies. And what I want to do is just go over the type of activities. When we talk about higher level thinking skills, we really want to get students to communicate and collaborate. Those are those 21st century skills. And I just want to lay this out so you can get a vision of what will happen once we go into the implementation of the lessons. So the very first lesson has students using a systems and critical evaluation of news articles. That's how they're actually introduced to the problem. They do that individually and then work together as a whole class. Then they go into the lab and the teacher has some demonstrations and the students work in small groups to actually understand carbon dioxide, to explore its properties, and to learn not only about carbon dioxide but about the carbon cycle as well. Then they come back together as a whole class to begin to define the problem of ocean acidification. They also at this point begin to learn about systems thinking, and then they move into individual work. Next, what they do is they come back together as a whole class when they begin to plan a cohesive set of experiments so they can make the best use of their time and so that they can model what's happening in current research. Once they work together as a whole class, they move into small groups to figure out exactly what their experiment will be. They then go into the lab to complete that experiment. While their experiment is running, there's individual and small group work with computers where they can supplement their particular experiment and their data with the amazing amounts of online data that is being put together throughout the world. Finally, they prepare for a global ocean acidification summit and then actually present in that summit and talk together about next steps and how this can possibly be mitigated. If teachers do all of these lessons, it generally takes 3 to 5 weeks of class time. And what I mean by class time is generally a student will be seen in a class for about 50 minutes 5 times a week. So that's the time that we're talking about. Why that 2-week gap? Well, that's because depending on how long students let their experiments run, that will definitely change. Teachers can do 1 or 2 of these lessons. In some cases, teachers do 2 or 3 of these lessons, and that works as well. So you can teach them individually or as an entire unit. Now, let's look at specifics for implementation. Now, when a teacher sits down to implement these lessons, the prior knowledge that students need to understand, or will do better if they understand, is the basics of networks. So we really want students to get to those higher level thinkings, and for ocean acidification, we want them to be a systems thinker. If they come in with some understanding, then they have a nice foundation for developing the systems thinking that's required to make decisions, not only about this, but about other sustainability and other issues in their life as well. We know that systems thinking enables behavioral change, and I encourage people, if you haven't been to this Waters Foundation website, there are so many wonderful resources on systems thinking. When I think about ocean acidification, specifically, there's a lot of habits I definitely want students to understand, and one of those is to consider both those short and long-term consequences of their actions. I also want them to consider how their mental models affect current reality in the future. I want them to find where unintended consequences emerge. All of these things, I want them to identify a circular nature of complex cause and effects relationship. There's a lot that we want them to do. And luckily, since all 4 of our modules that have been created deal with systems biology, we know that this basic understanding of networks is important. So we have 2 lessons that are available on our curriculum website, which I'll show you in a little while, that bring this basic understanding to students. Now, the way that we introduce this is through a classroom exercise where students analyze a social network. And this social networks comes from these fictitious phones that are laminated cards that each student has. And what the students do is it's a great icebreaker activity, is they walk around the class and they figure out, okay, if this is me, Claudia, who can I call in the class? And I walk around and I figure out, okay, 3A is Monica, 1E is Laura, and I write it all out, and then we come together. It's a jigsaw activity. We come together in classes and we define who can call whom, and we start to write it in the network. We call these circles in systems nodes. And whenever there's a relationship between 2 nodes, we draw a line, and if that relationship has a direction, we draw an arrow. So we call this an edge. This node can call this node, but this node cannot call this node. And this activity is modeled after real network rules that occur both inside our body and in ecosystems. As they pull together this network of 35 different nodes, they realize that they can do it, but it's kind of tricky. If you ask them to analyze it, if you give them a piece of paper with questions that says, okay, so what if 4A is grounded and 1A hears about a pop quiz that they'd like to share with the whole class? Will everyone know about it? And it's hard to ask these questions. And what we do then is we show them you can use tools to solve your problems. And that's what we do in systems research, is we develop these models and these tools where we can quickly perturb the network, make predictions about what happens, and then quickly test our predictions. These 2 lessons are available to you, and I'll show you where you can find them. And what these do is they build a nice foundation for the actual module. Okay, so now going into the module, Ocean Acidification, a systems approach to a global problem. In lesson 1, when students are first introduced to this problem, we didn't want to introduce it as a case study because we found in asking questions of students, we found that they were really tired of people telling them that the world is in a bad shape and they're the ones that have to fix it. And so we didn't want to do that. What we wanted to do is we wanted to present interesting case studies. And so what we did is we went to news media, and we have 41 different articles within this lesson. And each student reads a different article. And as they read the article, we give them a reading guide where they can begin to understand how you figure out the author's motivation and how you can kind of break down that article to know whether or not you can trust that resource or not. And then what we do is we have them find keywords, and we have them develop a small concept map where they can figure out, okay, what nodes are connected. So this is a fictitious article that is not part of the ocean acidification curriculum, but I wanted to include something here that wasn't related to any of the articles. So for instance here, they read an article about a saltwater lake that had some copper in it. They also know that halobacteria, it doesn't matter if I know what this is, but this stuff called halobacteria grows in the saltwater lake, so does brine shrimp. My article doesn't say anything about these two things, this halobacteria and brine shrimp being connected, so I'm not going to draw a line. So they go through this and they map out that network. And then the teacher brings the class back together at the front of the room and says, okay, as quickly as you can, each one of you is going to give me your concept map, and we're going to see if any of your articles are connected. And the reason we want them to do this is because we want them to understand the broad reach and accessibility of ocean studies. We want them to see how important it is, and we want them to gain the critical thinking skills to properly evaluate the news media that they're reading, whether it be online or in a newspaper. So what I'm going to show you here is a very messy diagram, because I'm going to show you that if you do this really quickly, that's all that you need to do to give it some meaning. So here's one that some students came up with. And what you see here is that every time a new word is mentioned, we put a little tick mark next to it. So here you see OA, and there's a little tick mark next to it. And every time something is mentioned again and again, we give it weights. So we can begin to see where the key nodes in this system are. Now your students may want to start arguing about it. They may say, okay, mine mentioned global warming, mine mentioned climate change. I think they're the same thing. You don't want to get caught up in that. What you want to do is get everything up there you can so that you can stand back and look at the picture. Now since this is such a mess, I'm going to show you a cleaned up version just between us. You don't need to do this in the classroom. But in the cleaned up version, if you put weight according to what nodes are mentioned time and time again, you begin to see this network. This network was made using Cytoscape, which is the same program we showed before with the cell phone simulation. But now you can ask students, you can say, okay, is this happening in today? And they say, yes, all these articles are fairly recent. It is happening today. Is any of this interesting? Yes. Here we are in a science class. Would you like to study these things? We're trying to learn about science and life. Yes, I'd love to study about this. Well, if we were going to study it, we have 180 days together in the classroom. What should we spend our time studying? Should we spend our time studying baleen whales? Well, that might be fun, but we can't really get them into our classroom, and they're not necessarily a key node. What's a key node or an important part? And then they say, look, this thing called CO2 comes up again and again and again and again. And depending on your students, some may know exactly what CO2 is, and some may not. Some may say to you, no, we cannot study CO2 in our classroom because there are alarms. And if there's CO2 in the air, that alarm is going to go off, and we have to get out of the room because we can fall asleep and never wake up. Oh, that's carbon monoxide. This is carbon dioxide. We have it. We have access to it. We can get plenty of it, and it seems to be important in our world. So let's spend some time going into the lab to learn about it. So then what they do is they go into the lab and they learn about CO2. There are two options. One is to use open inquiry, and the curriculum walks you through how you might do that. But what a lot of teachers want to do is they want to save that open inquiry experiment for later when they're really studying actual ocean acidification, and they want them to understand, to have more directed understanding of CO2. And for that we have these round-robin labs where they learn about cellular respiration through yeast and sugar. They learn about combustion reactions from burning different things. They learn about chemical reactions that release CO2, like baking soda and vinegar, and they learn how to detect it. And they learn how to detect it on a very low-scale way using lime water and pH as a proxy. And then they move through these round-robin labs. Now when I set this lab up, I always have at least 2 stations of this in my classroom and multiple stations of detecting if possible. So what I want to do is I want to take you to our website where we can see what some of this looks like. Now, if I can ask you to not follow along with me on your website. And the reason why I'm saying that is if we have 200 or so people all try to download one of our models, I'm worried of what might happen to our website. And I'm also going to be moving really quickly through the links because I can see I'm already running low on time. I'm going to move quickly through the links, and if you can just follow along on your screen of where I'm clicking, and then you can use this video recording in my PowerPoint later to move you more clearly through the website. Also, we're going to spend a significant amount on the time on our website, so you should be able to get an idea of where to look. Now, I have linked to this bit.ly tag, but if you search Ocean Acidification Systems Biology, you will get to our site. If you throw any other good words in there like curriculum or Montecoriana or Delega or Institute or Claudia, you'll get to our site. But I'm going to take you to this bit.ly tag, and that's going to open our main systems education website. Now here, I like to go to our modules menu. We can also get to our module through these curriculum modules pages. Here we have a description of all of our curriculum, but here's our ocean acidification, and we end up right at our landing page here. Now at the very top of this, there's the overview, a curriculum summary, teacher guides for people who haven't gone through this training or the training that I do in conferences. We also have that at-a-glance lesson outline, more information, and then at the bottom of every one of our pages, we have quick links. The first time you do our curriculum, I highly recommend that you read through the intro all the way through to lesson 6 before you teach it. If you read through a lesson and teach it, read through a lesson and teach it, it's not as clear of what you're doing to the students. So here we have just gone through the intro, and so now we're going to read this as a book. Okay, I'm ready for chapter 1. Now here, when we go to lesson 1, this is the lesson plan. Here we have our objectives, and it says see standards addressed. So when you go to standards addressed, here's our module, and you can see the links to Common Core Science Standards, Math Standards, Washington State, the Next Gen Science Standard, and the Ocean Literacy Standards. When you click on this link, what you'll get to is you'll download a document, and you'll see this spreadsheet of which standard is covered in which lesson. So this is a really useful thing. In addition to this, what we have is, because some of those standards, the wording can be unclear, we also have in our own words what students learn and what students do. So now as we go down, there's always our background information. Here's how you get to those cell phone lessons that I introduced. Here's a pre-assessment hard copy. There are survey monkeys for the pre-assessment as well. These are wonderful because not only can they help you, we can send you a report, but also they help us restructure this module. Here's that one article for each student, the reading guide. If you don't have time to read all 41 of those articles, you can click on this link, and what that does is it downloads a summary of each article for you to use, so you can just read a summary and then you can know which article to give to which student. Always at the bottom we have our quick links of whatever resources are available, and then we have some really great accommodations depending on what type of students you have and what type of resources you have. And then we also have our extension activities. Okay, now lesson 2 is when they go into the lab and they explore that CO2. So again, we have all of the basic information up top, and then our 2 recommended options, and then we have our 2 demonstrations. One demonstration is a visual pH scale. Now, if your students don't have much pH understanding, also built into this lesson, we have a lot of different pH activities. What this pH scale does is it shows them a nice picture using universal indicator of the various range of pH. And then what the teacher does is take some CO2 that's been produced and hovers over those beakers, and you can see the pH drastically changing just from that CO2 that's being released near those beakers. The other demonstration that is a good jumping off point is this balloon drop, where you take 2 balloons, one filled with carbon dioxide and one filled with just air, and then you hold them up high and ask the students what would happen if you drop them. You let them both drop and you will see that CO2 balloon reach the floor first. We have found that this gives a terrific visual for students to imagine CO2 going into the air and falling right into that water. We notice that students have a difficult time. They think, oh, well, here I am. Let's say I live in Chicago. The CO2 that we're releasing here, there's no way it makes it to the ocean. But we know that's not true. We know the globe is connected through weather. We know the whole ocean is connected through a conveyor belt. We know a lot of this, and having visuals like this really helps students to begin to understand that. Here's our round robin labs. Now, one of the things that we always have with all of our curriculum is everything that can be in a Word document is in a Word document, so that you can click that enable editing and you can change it according to your students. So if you wanted to work in stoichiometry here, you would be able to do that. Depending on what you want to work in, you're able to do that. Here we have pictures of each step, and students can write their observations here. Some teachers like to laminate these and put them at the station, and then they can use those boxes. They can write those things in their lab notebook. So here are those pH resources that I mentioned to you, and also a little bit on how we use logarithmic scales. That's important because what students don't often realize is that a 0.1 change in pH is a 30% or so change in acidity. So that's something that we like them to really try to understand. And so these resources, that project at the University of Colorado is fabulous with what they have there. Specifically to using a claim evidence reasoning at this point in the curriculum is wonderful because by the time they get to their ocean acidification experiment, then they can really be proficient in those and they can use them for their summit. Okay, so now lesson 3. The next lesson is when the students begin to define the problem of ocean acidification. And the way they do that is they come back from the lab into the class together. And this 20-minute acid test, the Global Challenge of Ocean Acidification, is a video that was created by the National Resources Defense Council. And it's a fantastic video that really helps students see that this is a global problem. It's not a problem for people living just on the coast or in island nations. It's a problem with many stakeholders. It also helps set the stage to model the collaboration that is needed for ocean acidification resource and really is needed for many of our big scale problems that we're facing today. The other thing that lesson 3 does is it really begins to build in the idea of a systems study. It gets students thinking about the system, figuring out if there are many parts with a lot of interactions, if there are any emergent properties or things that come about that are surprising, and if there's any reverberating effects, which are these indirect effects that happen several steps down the line. And one of the things I want to share with you is how we really get them to see this. We always start with students with something that they already know. And at this point in the curriculum, they know that animals respire. They release the CO2, and trees and plants take that in. They do photosynthesis, and then they give oxygen. And then we ask the students, does this seem to be something that's balanced? And in many cases, we don't want to argue too many points, but we say, yeah, it kind of seems to be balanced, right? There's some checks and balances in place. It seems to be balanced. Well, does this also happen? Then we also say, does this also happen just on land or in the water? And they say, oh, it happens in the water too. I read that in my article. It does. So we have marine animals that also respire and release CO2. We have phytoplankton and plants of the sea and other things doing photosynthesis. They take in that CO2, they release oxygen not only to those marine animals, but they also release oxygen for us, a lot of oxygen. We think of those phytoplankton as kind of one of our lungs, and the other lungs being what's happened on land. So we know that this is taking place, and we ask the students again, does this seem to be in balance? And in balance, and they say, it seems to be, and we don't want to get into the finite pieces of this. We say, yeah, again, I've heard about diatom blooms. I know it's not always in balance, but it seems to be. And we say, okay, since the Industrial Revolution, we have a new respiring group. Let's call them combustion reactions, the burning of fossil fuels, whatever we've needed since the Industrial Revolution. What's their counterpart? And this is where, as a teacher, we give them lots of wait time. We don't tell them the answer, right? As you say, what is their counterpart? And always there's at least a few students in the class that say, is it the ocean? And then you say, yeah, what do you think? Kind of seems to be. What is big enough to be a counterpart to all these combustion reactions? Oh, it's the ocean. So what does that mean? Is that a good thing or is that a bad thing? What does it mean for all of these folks? What does it mean for these? What does it mean for these? I don't know. That's what research is about. And this is where we always get students hooked. This is the point in the curriculum where they are, oh, wow, so here's somebody not telling me what's right or wrong and what I have to fix, but they're telling me, oh, this is why we do research, because we want to answer this question. We want to know what it means. Well, all right, let's go into the lab and do some research. But before we just jump into the science, because this isn't just a scientific situation, right? It has impacts on society. So then we say, all right, well, since it has impacts on society, let's align ourselves with an interest group. Let's align ourselves. Let's say that we're somebody who needs these combustion reactions because, let's face it, there's a reason we use so much energy. Many of us need it and require it, or so we think, right? So each student then writes on a small piece of paper, if they were going to align themselves with one of these groups, with perhaps the group that is a high CO2 emitter, or perhaps the group that is a low CO2 emitter that doesn't need a lot of energy but does rely on the health of their ocean for their livelihood. Or what if you're these diatoms that use that CO2? Are you a winner in this situation? Or what about these marine calcifying organisms that we know if there's a pH change that they're not able to make their shell in the way that they can? What about them? And we ask the students, if you were to advocate for someone, who would it be? And so the students write on a small piece of paper their first choice and their second choice, and then the teacher comes back together and then puts groups together, puts those photosynthesizing groups together, the low CO2 groups together, and then these become their lab groups. So now the students then sit down together and they begin to plan their experiment, not just for the science, but also for their interest group. So lessons 4 and 5 of the curriculum, 4 are students planning their experiment, and you need to give a good amount of time for them to plan their experiment. If you do give a good amount of time, their experimentation goes extremely well. And then lesson 5 is them doing that experiment. Now the main question that everyone will study is, what effects does this increasing atmospheric CO2 have on the ocean and its subsystems? Now this is the umbrella question. Now based on the group's subsystem, they'll narrow that question. And they'll narrow that question until they brainstorm and get to a point of what they want to do. Do they want to try to grow snails? What is their group? What's going to give their interest group the most information? We want them to model collaborative systems research. We want them to use multiple stressors. And the way that they can use multiple stressors is if they have multiple and diverse data types. And I'm going to talk to you specifically about those 3 things in the next few slides. Now, first, just a word on multiple stressors. We do a really good job teaching students that when you do an experiment, you change one variable and you control everything else. And that's a wonderful first step. That's how we begin to see cause and effect. But in the real world, especially with something like ocean acidification, we know that we cannot decouple certain factors, even CO2 and pH. We know that organisms live within a very narrow range of pH. And if you push them outside of that pH, you stress them out and perhaps even kill them, right? But you do that while you're also adding CO2, which has other factors. So the next step for students is to begin to think about baby steps towards moving to these multiple stressors reactions. And let me talk and show you how you can do this. So I'm showing you a nice picture that my interns did, but we've replicated this in classrooms and it works really well. So this is a group that is, their interest group is the photosynthesizing non-calcifying organisms. And so what they use here is Thalassioscirus pseudonana, which is a diatom that uses silica for its shell. We'll call it FAPS. And so they grow FAPS in all the nutrients they need with high light. And then what they do is, and normally you would see 3 different replicates here, but for just a picture's sake, we took 1. They also grow them with phosphate-limiting media, silica-limiting media, and nitrogen-limiting media to show how the impacts of upwellings and to teach more about biogeochemical cycling. And what the nice thing here is that you can see this. So if you don't have time for your students to count cells, or if you don't have microscopes, you can see visible differences. You can see here these cells do not like if they don't have nitrogen, and even if they have all the light in the world, they still don't grow very well. So here's what one group has time to do. Well, what if there's another group that is interested in studying something similar? What they want to know perhaps is they want to show that the blooms are important for these low CO2 emitting nations, and so what they might want to do is a similar thing. And these groups then get together, and what they do is they realize, okay, so if you're going to do high light with all these, let me do low light with all of these. And all of a sudden you're adding different stressors or different variables onto your experiment. Another group can then go ahead and say, all right, I'm going to change CO2. I see you did this in ambient current level CO2. But what I'm going to do is I'm going to add some CO2, and then we're going to see what happens. And then they collaborate and share their data to get understanding. Now, as I mentioned, you can just see the difference in these, but you can also get very technical with these. And I'm going to show you some pictures of what it looks like on a nice technical scale, because it's always easy to back down from that. So in this example, students combine their data for one graph, so you can really see how the cells grow. They used hemocytometers to do this. If you don't have hemocytometers, what you can do is you can just use slides, gridded slides, and the curriculum actually walks you through all of this. In addition to this, what if your cell counts are the same, but your cells look funny? They're kind of misshapen. Well, then you don't have very healthy cells. So sometimes cell counts don't give us all the information we need. We want to do a pigment extraction, perhaps, or a pigment description, or a cellular description. We have many different ways, and depending on which cells we use, we can get different types of data. And when we have multiple and diverse data, that's where we can tease out what's happening with those multi-stressor experiments. Now, what students are encouraged to do is make small variations in their experimental procedures. That's what helps them facilitate that collaboration. So they can do different water types. So they can use freshwater or marine water. Of course, we don't want them to try to grow organisms in freshwater that aren't suited for that. But the marine water, they can use aquarium water, which is just a powder that they make. They can collect it from the environment. They can make it in the lab. All of this how-to material is on our website. They can use filtered or not. They can change various nutrient levels. They can really do all of this in small variations to make it not a logistic nightmare for you as a teacher, and then also to give a lot of understanding. As far as CO2 source, they've done those carbon dioxide experiments, so what they realize is I know lots of ways of making CO2. I can use my own CO2, I can use yeast, I can burn different things, chemical reactions, dry ice. There's all sorts of different things they can do and different ways they can transfer that CO2 through tubing. So here's an example. Again, what I want to show you is this is a more high-tech one, because you can probably see how you can make this more low-tech. So this is a Vernier Lab Quest, and here what we have is we have a container, and we put some dry ice in this container. This is a carbon dioxide probe. This is a pH probe that's inside this marine water, and there are shells in here. And so then the students took out their shells and they massed them, and they noticed that the shells left in that seawater lost 2% of their mass over 3 days. And you can see how it begins to be an engineering problem. The students notice, even though I've got these fancy O-rings, I'm still losing some CO2 mass. So they tried aluminum foil and they realized, oh, CO2 even goes through aluminum foil. That's interesting. So they try different things. I've also made these containers out of big jelly bean Costco jars where you can just cut out and you can use a pH test strip. There's a lot you can do with these experiments. Now, your students in the lab, some of them will need to be in the lab for that entire 50 minutes every day, counting cells, with a really intense lab component. And some groups will be in and out of the lab in 5 minutes where they mass their cells and they're done. And so to replicate what we do is make good use of our time, and then also logistically to make it possible in the classroom, we have these supplemental activities, an online data component where students actually compare their data and get more data online. And because we're running out of time, I'm going to just mention that all of this is in our curriculum. And these carbon footprint calculators, it's a good opportunity for them to go to those carbon footprint calculators and begin to get information not only for their interest group, but to compare models. We also have some models available where they can make hypotheses and test them. This model is not meant to be perfectly accurate, but it's meant to get students thinking about what's happening in the environment. Now, in the next 5 minutes before we kind of break for questions, I'm going to take you to back to our websites so I can point out just a few things. So we mentioned Lesson 4 is where they collaborate. This Teacher's Guide to Student Experiments doc is one of my favorite documents because what it does is it breaks down for you what the different interest groups can think about doing. So that's a really nice resource for you. Also, on this page is how to get them to plan, how to scaffold them through that, and then this Teacher Guide to Experiments, again, it helps you track this teacher tracking experiments. It'll help you track if you have 8 groups in each class and you have 5 classes, it can become a logistics nightmare. And so that tracking student sheet is really wonderful to use. Okay, in Lesson 5A, here's their actual experimentation. So we broke Lesson 5 down into experimentation in the lab and then also online component. So here are all the lab techniques you need. Here are some basic protocols for diatoms, some basic shell dissolution protocols and some guides. General chemistry, a lot of groups just want to do general chemistry. They want to know how low the pH of marine water, buffered marine water, can actually go. That's a good experiment. They don't have to use live organisms. Also, these are nice examples of these multi-stressor experiments that we talk about. Okay, so here is all of that, all that you need in there. Lesson 5B breaks it down into these online components. And here in this webinar series, we're actually going to be showcasing some of these online components. So here we have more in-depth carbon cycle learning. This virtual sea urchin lab is a terrific one for students that are interested in shell dissolution and what's happening to organisms that have that have calcium carbonate shells. Here's that model that I showed you. Here's different data sources around the globe and how they can be used for high school students. Also, this What the Past Tells Us is really something that's important to look at. Ice cores tell us a lot about what's happened in our history. They allow us to see that the rate of carbon dioxide change that we're currently seeing is about 100 times faster than observed. And so these are important big picture ideas that we want students to get out of it. Now once they finish those, they come back together and begin to plan for their global summit. Now this lesson is broken into 3 parts. We have advanced prep and we have some pictures that help getting students to think about what they'd like to present and what's presented at scientific conferences now. We give them time in class to prep. This can be done for homework, but there's a guide getting them to prep for both their presentation and the summit, and then they present their scientific posters, and then they come back together on day 3 to debate and come up with a resolution on atmospheric CO2. So each of these comes with guides for helping students see the big picture. Here's a picture of a teacher who brought the students back together and really getting excited about what was happening just by showing some nice pictures of what's happening around the globe. So the thing I want to emphasize with the final culminating activity is you really want the emphasis to be placed on the impact of the subsystem and the system. And what that means is you don't want students they have a habit of looking at their experiment and only their experiment. We want them to take a step back and see, okay, what would happen if your experiment was in the real world? How do you get them thinking about that system? And how do you get them reflecting on these unanswered questions and what their next steps would be and what their roles are given what they now know? And so one way that we do that is we give them some guides. And here what we have is we have our 4 different groups. These are our 4 different interest groups. And we have students kind of figure out what is the effect between these different nodes. If atmospheric CO2 level is increasing, we know that CO2 absorbed by the ocean is increasing, which increases carbonic acid, which decreases calcium carbonate, which has a negative impact on these marine calcifying organisms. And what we get students to do is see if there are positive or negative feedback loops within this system that will help regulate it. And there's a couple of different ones of these. Now, the last thing that I want to emphasize before we stop for questions is that when we field tested this over the last 4 or so years, we've been able to see what emphasis are most important. And we noticed that if teachers completed the cell phone lessons prior to this module, the students, when they get to that global summit, they focus more on reverberating effects. We see them having really high-level conversations where they don't just stay in the minutia and the details of their experiment, but they also begin to make predictions about what is happening on a grand scale. If they don't do those cell phone lessons, we see some difficulty in having them get to that point. We also notice that if in their experiments they use that systems level emphasis with multiple stressors, multiple and diverse data in collaboration, we see great gains both in thinking and in scientific inquiry. If they don't do that, and a lot of teachers do teach these lessons in 2 weeks, and what they do is they use the protocols that are online, and they have one variable change, and then they test what has changed in response to that one variable, we still see a gain in general inquiry and scientific thinking. So it's still worthwhile to do that way. So that the systems level emphasis is a bit scary. It's okay to start here and then maybe slowly bring in that systems level emphasis to get to those great gains. We also notice there are teachers who don't want to bring in the interest groups, and they don't want to spend the 2 or so days on that global summit. And when teachers don't bring in the interest groups, the students are still engaged. But when they bring in the interest groups, we see a lot of passion. And not only do we see a lot of passion, but we see a global perspective of what's happening. And then the thing I want to end with is that students love this. I've heard time and time again that it's fun, and from this particular quote comes from a student who was not having fun in school at all, and he said to me that this was the most fun I had ever had in a science class, and this is, you know, as a junior telling me this. And we hear time and time again from the field test assessment that they wish they had more time. They really liked their experiment. They really wanted to take more data. They really wanted to see what happens. So now we're going to break for questions, and just as Laura and Jen kind of get ready for questions, I want to thank Monica Oriana and Nitin Baliga. Without Monica's research, this wouldn't have been possible, and everyone in the Baliga lab has been incredibly supportive, and everyone at ISB. And in addition, the effective practices for communication ocean acidification, this SOARCE group has been wonderful to work with. And without the funding from the National Science Foundation, these modules never would have been developed. And without the Center for Systems Biology, which is a National Institutes for Health center, then the dissemination that takes place of these modules would not be as great. And of course, our curriculum team, these are all of the teachers and students and scientists that have spent a significant amount of hours giving us feedback, writing the lessons, and I'm just so pleased and happy to have been a part of this, and I'm very thankful for everybody's hard work and expertise.
Thank you so much, Claudia. That was a fantastic presentation, and I know I've learned so much, and it's such a comprehensive resource. I can't wait to explore it more. So now we're going to open the floor for questions. So please type any questions or comments that you have into the question control panel. And I'll go ahead and start with a question from Patricia I'm not sure I'm going to pronounce this right, Azinhaga. And she asked, are the articles in Lesson 1 scientific articles or articles that use simple language?
Can you repeat the question? I'm sorry.
So the articles that you mentioned that the students are reading in Lesson 1, are they scientific articles or are they articles that use more simple language?
They're not scientific articles. That's a terrific question. So they're all from news media, either online or even blogs in some cases, and they are from very accessible. We aimed them towards 9th grade reading levels. And so there is a resource list for scientific articles. Some teachers really wanted them to dive into a primary source paper. We do have those available, but in Lesson 1 they are all news articles, and they are all short enough for them to read and analyze within about 30 minutes. But some of them are longer than others, and that's kind of the teacher's job to pass them out to the student that they think would be best for that particular article.
Great. I have a question from Victoria Caro, and she asks, is there one lesson that you would recommend teaching if you only have students on a field trip basis?
Yeah, I would, that's a good question. It depends on what my goal was. It really, if I'm thinking not of networks and systems, but if I'm thinking about ocean acidification, I think that I would, that's a tough question. I think I would, those experiments in Lesson 5, there's a lot of really good and interesting things that you can do, but you can't really teach that in one day because you need those cells to grow, or you need to have time for that coral to change, or, you know, I'd have to think about that a little bit more, I guess, is unfortunately my answer on that. Also, in Lesson 3, there's a lot that can be done in one day that has huge meaning. So I suppose if I had a short amount of time with students and I really had to pick something, I'd go with what is in lesson 3.
Okay. And there's a question from Kelly Boyd. Are the questions more appropriate for middle or high school?
High school.
Okay.
We have 8th graders that have done this, but I'm not aware of any 6th or 7th graders that have done it. I received emails from teachers who said they were going to try it with 6th or 7th graders, but I never heard back from them. But I do know of 8th graders who have done it successfully, and we really try to write it towards 9th and 10th graders.
Okay. I have a question from Mary Hunterly, and she said, why not include information about decomposition in the ocean balance discussion?
What a terrific thing. Yeah, I think that would be great to include.
Excellent.
In the carbon cycling lessons, we do have some PowerPoints on carbon cycling. We do show a little bit of that. There's some nice graphics and depiction of what's happening with decomposition and how it contributes to the carbon cycle. So we have a little bit in there, but it's not stated. You can certainly build it out.
Okay, I have a question from Nancy Flowers, and she's asking, what are the requirements for culturing the diatoms, and is there any special equipment needed?
No special equipment needed. It's nice to have tissue culture flasks because they're sterile and they grow so nicely. You don't have to worry about other things growing in there. But if you don't have them, we have plenty of teachers who use just glass flasks, and they rinse them out really well. And if you have an autoclave, that's fantastic. But we do have teachers who do this without autoclaving them, but you can't do the experiment for as long because other things do tend to start growing in there. But they're not dangerous things that grow in there. The thing that you really do need, I find, to make it meaningful are microscopes. If you can get them to look at the cells and count the cells and see the cells as they're, and we even have pictures of them on there. If you have microscopes, that's ideal. You don't need micropipettes. You can use disposable barrel pipettes. So I think microscopes are the most important thing that you need.
Excellent. I think that's about all the time we have for questions, and just want to thank Claudia again and thank all of you for joining us. And we welcome any feedback, further questions, or suggestions for topics for the webinar series. If we didn't get to your question, we'll send the question log to Claudia, and we'll try to get the questions answered within the next couple of weeks. You can contact me, Laura Francis, or Jen Bennett. Our emails are listed if you'd like to, if you have further questions or suggestions for future speakers for the series. And our next webinar is going to be on May 14th, 2014 at 6 p.m. Eastern, 3 p.m. Pacific. And we'll have Meg Chadsey and Paul Williams, and they're going to be talking about ocean acidification, a Washington State curricular framework. And you can go to the website to find out more information about that. So thanks again for joining us. And we're going to go ahead and end the webinar, but please continue to ask us questions, and we hope to see you on a future webinar. Thanks again.